Imagine your body as a vast library, stretching as far as the eye can see. Each book on the shelves is a set of instructions. Your chromosomes, carefully written and stored in every cell. And within those books, each sentence is a gene, a unique line of code that helps build and run your body. These genes decide everything: your eye color, your height, the way your hair curls, and even how your body responds to the world around you. They control the rhythm of your heart, the strength of your bones, and so much more. This entire collection of genetic instructions is called your genome, the ultimate manual for making you, uniquely you. But sometimes in this massive library, a tiny typo sneaks in, a single letter out of place in a gene. Most of these typos are harmless, like a small spelling mistake that doesn't change the meaning of a sentence. But some typos change crucial instructions, leading to genetic diseases like cystic fibrosis or sickle cell anemia, which can affect lives in profound ways. For centuries, medicine could only treat the symptoms of these conditions, offering relief but never truly addressing the root cause, the typo itself, hidden deep within our DNA. But now, science is changing that story. We're learning to become genetic editors, using powerful new tools to read, understand, and even rewrite our genetic code. Enter gene therapy, an approach that doesn't just manage problems, but aims to fix the source, correcting the instructions themselves. Imagine a magical pen that finds and corrects the misspelled word in your genetic book, restoring the sentence to its original meaning. The goal? To fix the manual so your body can work as it should, giving people a chance at healthier, fuller lives. This isn't science fiction. It's happening now, offering real hope to families worldwide and changing the future of medicine. The possibilities are just beginning, and the next chapter in the book of life is being written right now. So what exactly is gene therapy? It's a revolutionary approach in modern medicine that's changing how we think about treating disease. Think of it as a biological software update for your body, an upgrade that goes right to the source of the problem, your genes. Instead of just patching up symptoms with medication, gene therapy aims to correct the underlying genetic code, your DNA, where the real issue begins. The ultimate goal is to fix diseases at their root, not just manage them over time. This means targeting the cause, not just the effects. Imagine a one-time treatment that could actually cure a genetic disorder, rather than simply controlling it with lifelong medication or therapy. This is especially powerful for diseases caused by a single faulty gene, like hemophilia or muscular dystrophy, conditions that have been difficult to treat until now. For many patients and their families, gene therapy means the chance for a life free from daily struggles, hospital visits, and constant worry. But it's a complex process. Scientists must deliver the right gene to the right cells, safely and precisely, without causing harm or unwanted side effects. It's not as simple as injecting a new gene. It requires a deep understanding of genetics, advanced technology, and careful delivery methods to ensure success. With every breakthrough, gene therapy becomes more precise, more personal, and more effective for each individual patient. It's the cutting edge of medicine, treatments tailored to your unique genetic makeup, offering hope where there was little before. Science at its most hopeful, opening new doors for patients and families around the world. Like a mechanic with a toolkit, scientists have developed sophisticated methods to repair and enhance our genetic code. These methods can be categorized into 4 main strategies to fix genes. First, gene replacement. If a gene is faulty or missing, a healthy copy is added to do the job. This can restore normal function to cells that were previously impaired. Second, gene repair or editing. Tools like CRISPR act as molecular scissors, correcting the typo right where it happens. This precision allows for targeted fixes without affecting other parts of the genome. Third, gene addition. Sometimes we add a new gene to give cells a new function, like helping immune cells fight cancer. This can enhance the body's natural defenses. Fourth, gene silencing. If a gene is overactive or harmful, we can turn it off, stopping the production of a toxic protein. This is particularly useful in conditions where gene overexpression is a problem. Each approach targets a different kind of genetic problem, offering a tailored solution. Gene replacement is common for missing proteins, providing a straightforward fix for certain genetic disorders. Gene editing is precise, fixing the error at its source, ensuring that the correction is permanent and accurate. Gene addition gives cells new abilities, like supercharging the immune system to better fight off diseases such as cancer. Gene silencing dials down harmful genes, especially in neurological diseases where overactive genes can cause significant damage. These strategies let scientists tackle genetic diseases from every angle, providing a comprehensive toolkit for genetic therapy. The toolkit is flexible, powerful, and growing, with new tools and techniques being developed constantly. With each advance, more diseases become treatable, offering hope to millions of patients worldwide. The future of medicine is in the genes, and the possibilities are endless. So, how do we deliver healthy genes to the right cells? Enter the vector, the delivery vehicle. The most common vectors are modified viruses. Scientists remove their harmful parts, turning them into harmless delivery trucks. The healthy gene is loaded inside, and the virus naturally delivers it to target cells. Different viruses work for different jobs. AAVs for nerve or muscle cells, lentiviruses for long-lasting effects. Choosing the right vector is crucial for safety and effectiveness. Non-viral vectors, like liposomes or nanoparticles, are also being developed for safer delivery. Each method has trade-offs. Viral vectors are efficient, non-viral ones can be safer. The science of vectors is evolving fast, aiming for the safest, most efficient delivery. Getting the gene in is half the battle. Doing it safely is the other half. Once the gene is ready, how do we get it into the patient? There are two main methods, in vivo and ex vivo. In vivo means delivering the vector directly into the body, ideal for organs like the eye or muscles. Ex vivo means taking cells out, fixing them in the lab, then returning them to the patient, common for blood or immune cells. In vivo is simpler but less controlled. Ex vivo offers more safety and precision. Ex vivo is used for blood disorders like sickle cell anemia, building a new healthy immune system. Each method has its place, depending on the disease and cells involved. The right approach can mean the difference between managing and curing a disease. Gene therapy is already changing lives. Take severe combined immunodeficiency, bubble boy disease. Ex vivo gene therapy lets children build new immune systems and live normal lives. Sickle cell disease, once a life of pain, is being cured by editing blood stem cells with CRISPR or adding healthy genes. Patients are now free from pain and producing healthy blood cells. Gene therapy has even restored sight. For Leber congenital amaurosis, a one-time injection into the retina has helped people see stars and read for the first time. These breakthroughs are the result of years of research and brave patients. They prove we can do more than manage symptoms, we can offer real cures. From immune systems to eyesight, gene therapy is turning the impossible into reality. Each success brings hope to millions. The frontier of medicine is here. Despite the promise, gene therapy faces big challenges. Cost is a major barrier. Treatments can be extremely expensive. Making these cures accessible to all is a top priority for the future. Safety is another concern. The immune system can react to vectors, and there's a small risk of disrupting other genes. Long-term effects are still unknown. We need decades of follow-up to be sure. Researchers are working to make therapies safer, cheaper, and more precise. The list of treatable diseases is growing. Hemophilia, Huntington's, even some cancers. Gene therapy is more than medicine. It's hope for a healthier future. As we learn to edit genes, we're also discovering new layers of control, like epigenetics. But that's a story for another time.